Hi, everyone, and welcome back. In this video, I'm going to be talking about chromosomal abnormalities and extranuclear inheritance. So what happens if we end up with an improper number of chromosomes? And we'll also talk about how certain things get inherited, particularly mitochondrial diseases can be inherited through extranuclear inheritance, which means the DNA is not contained within the nucleus. So let's get right into it. So the first thing we'll talk about is how we describe variations in chromosome number. Okay, so there's two ways we can talk about this. We can talk about it in the context of aneuploidy, which is the loss or gain of one or more chromosomes, but not a complete set. So this could be something like monosomy, which is the loss of a single chromosome from an otherwise diploid genome, or trisomy, which is the addition of a single chromosome from an otherwise diploid genome. So perhaps the most well-known example in humans would be trisomy 21. Those are individuals who have Down syndrome. So they have 3 copies of chromosome number 21. Okay. It's not a complete set of chromosomes. It's just they have 46 plus 1, right? And we've learned how to write that when we do, in my previous videos, I discussed how we annotate that when we do a karyotype, okay? Or we can talk about polyploidy. And when we talk about polyploidy, we're talking about when there's, I say when there's more than 2 complete sets of haploid chromosomes. This all falls into a broader umbrella of euploidy, which is how many chromosome sets does an organism have, okay? Now, when we talk about humans, we are diploid organisms. So in the world of euploidy, we are diploidy. The proper number of chromosomes for us to have is 2 copies of each. And you should have 2 copies of chromosome 1 and 2 copies of chromosome 2, one from your mom and one from your dad. That is not true in all organisms, particularly in plants. So it turns out that polyploidy is not really well tolerated in animals, but plants seem to be okay with it. So make sure when we're talking about the variation in chromosome number, we know what we're referring to. Are we talking about the addition or loss of a single chromosome from a pair or the appropriate number? Or are we talking about an organism that has multiple entire sets, right? We have 2 sets of chromosomes, one from mom and one from dad. But if you look at a lot of our crop species, they are polyploid. They have 3 copies or 7 copies or 8 copies. Yes, there are octoploid organisms out there. It turns out that this seems to be a very important aspect of evolution in plants. I won't get too far down that evolutionary rabbit hole in this conversation, but just be aware. We tend to think of all organisms as being diploid because we're animals, and a lot of what we interact with are animals, which tend to be diploid. But particularly in plants, it is possible to be polyploid as a normal level of chromosomes. Okay. And as you would anticipate, changing the chromosome number can have very profound effects on whatever organism this happens in. So let's start off talking about monosomy and trisomy. So monosomy is, you can kind of annotate this as 2n minus 1, right? The 2n is the total number of chromosomes. So for us, 46 minus 1 would make us 45. You'd have 45 chromosomes. Basically, you'd have 22 complete sets and then 1 set that's like, I only have 1 copy. This is not particularly well tolerated in humans. The one exception that seems to be compatible with life is Turner syndrome. These are individuals who are genetically female. They have 45 chromosomes. They're missing 1 copy of the X. So they only have 1 X chromosome. And while this is tolerated, I should say is compatible with life, it's not without an effect. So individuals with Turner syndrome, depending on the severity of it, can exhibit changes to their anatomy and their physiology. And when I'm talking about the severity of it is this. If you are a genetic female carrying 1 X chromosome, sometimes that other X chromosome isn't lost in every cell. It gets lost in a cell lineage, which means they are mosaic for that, which means some of their cells have the proper amount of X chromosomes, But in some of those lineages, they've lost one copy of that. So some of the cells only have one X chromosome. And as a result, only some of their cells have that sort of X deficiency, if you will. And they get the fewer number of cells lacking an X chromosome, the less profound the impact is on the individual. But some people are just flat out missing one of their X chromosomes, and they have they will have the most observable phenotypes as somebody with Turner syndrome. We think monosomy is likely fatal for one of two main reasons. The first is that if there is a lethal allele, so at this point, this person is hemizygous for that particular chromosome, right? Whatever chromosome it is, they are hemizygous, which means they don't have a second copy. So if there is chromosomes have thousands of genes on them. If there's a single mutation on a chromosome that's unpaired, that could be lethal. Well, there's nothing to compensate for it. And as a result, that particular chromosomal arrangement will not be compatible with life. We talked about that a bit when we talked about X-linked inheritance in males, where they don't have that second X chromosome to balance out any sort of mutations on the X chromosome, things like hemophilia, things like color blindness. Well, in this case, what we're talking about is having a very similar situation, except one of those alleles is actually fatal. That whatever mutation or version of that gene is not compatible with life. And you would count on a second copy of that, a healthy copy of that gene to balance that out and rescue the individual. But when you don't have a second copy to balance it out, that fatal allele will be the one that gets expressed. And as a result, it's incompatible with life. The other is something called haploinsufficiency. So this gets into gene dosage. So sometimes for some genes, we need two functional copies of that gene, because that's the only way to produce enough of the gene product, usually a protein, but enough of that gene product for the individual to survive. So just the sheer absence, even if the copy of the gene that's present is compatible with life, the amount of protein or gene product produced from it isn't sufficient in and of itself to allow life to exist. You need both copies to get the right amount of gene product for normal bodily functions. Okay. The one thing that we need to point out when we're talking about monosomy, trisomy, any of these things, we are talking about things that typically happen during gamete formation during meiosis. So the one thing I want you to keep track of when we're talking about chromosome number, when we're talking about these defects that are happening, this is all happening during meiosis in one of the parents. Okay. The impact is going to be on the forming zygote. It's going to be what happens when that malformed gamete fuses with another gamete to produce offspring. But the defect is originally occurring during the formation of the sperm or the egg in the parents. Okay. And one of the things we know is when these mutations occur, it seems to be better tolerated in the plant kingdom as are most other chromosomal alterations. Okay. A lot of what we're talking about is not going to be something that's compatible in humans, but since genetics impacts all living things, we have to talk about this in the context of all living things, including plants. So again, monosomy, not particularly well tolerated in humans, with a few notable exceptions. What about trisomy? So trisomy is 2n+1. So for example, trisomy 21, which I mentioned, this would be written as 47 21 plus. So this is somebody who has 47 chromosomes, means they have the normal 46, two copies of each, plus an additional third copy of the 21st chromosome. And this is the most common. 1 in 800 live births in the United States will result in an individual with this particular version of trisomy, which then the phenotypically is described as Down syndrome. Typically what happens that we found is there is this non-disjunction of chromosome 21 during meiosis in anaphase 1 or 2. Basically, what happens is when these chromosomes are supposed to split apart during anaphase, either during anaphase 1, during meiosis 1 or during meiosis 2, they both go in the same direction. And as they go through either the second round, or if it is the second round of chromosomal separation, you're going to end up with a gamete that has an inappropriate number of chromosome 21s. So when that then goes on to fuse with the other haploid gamete, now all of a sudden there's 3 copies of that in the resulting zygote. And you can see this is what it looks like. So if it's meiosis 1, where the non-disjunction happens, you will see that the resulting gametes will have 1 copy of dad's chromosome 21, 1 copy of mom's. These would be the grandparents. And then when this goes on to fuse with the next gamete, you'll get a 3rd version of it. If it happens during meiosis 2, you can see that the trisomy results in 2 identical copies of the 21st chromosome, making it into the gamete, okay? The other thing to note is that when this is happening, you will also note that there are gametes that lack chromosome 21 altogether. So again, it's not like we're creating additional copies. Some of these gametes will lack chromosome 21 altogether. So this particular gamete right here would only have 22 chromosomes in it. It would be missing the 23rd chromosome or 21st chromosome to make it 23, right? When this fuses with another healthy gamete, these are going to be non-viable. These will result in non-viable pregnancies in humans because, quite simply, there's just not enough chromosome. Remember, this would be a great example of monosomy at this point. Since monosomy 21 is not well tolerated, these would likely result in what's called spontaneous abortion or miscarriage. This is what a perfectly normal way for a body to decide that this is a non-viable fetus. We're going to have to try again. All right. So these are 2 different scenarios in which this happens. It's very similar for the other versions of trisomy. So there's something called Patau syndrome. This is when the triplicated chromosome is going to be chromosome 13. There's Edwards syndrome, which is 3 copies of chromosome 18. Both of these happen and can result in live birth. That being said, in both of these cases, there will be severe malformations, both physically, there will be major problems cognitively, and in all cases, individuals with Patau or Edwards are going to die very, very early on in life. Think weeks to months to very few years in some cases. In many cases, just because the impact of having 3 copies of these chromosomes is just so profound. And the one thing that we have to rectify, and you may have heard this if you're someone who's had kids or someone who is trying to have kids, that a significant number of pregnancies in humans result in spontaneous abortion. And in some cases, the women who are pregnant may never even know that they were pregnant because it happens, and it just happens in conjunction with their next period, and they just think, oh, this is just I'm just menstruating, and actually what it is the body getting rid of a potential pregnancy just because there are chromosomal abnormalities. And the estimates are right around 6% of all conceptions will have some instance where there's an abnormal number of chromosomes for whatever reason. It doesn't have to be these ones we're shown here, and it results in spontaneous abortion. Now, polyploidy is an entirely different animal. We're not talking about missing or adding in a single chromosome. We are talking about the addition, or in some cases, subtraction of entire chromosome sets. So what you have to think about, since we're a diploid organism, you really have 2 entire genomes inside of your cells. You have 1 whole genome that comes from your mom and 1 whole genome that comes from your dad. And that makes us diploid. But very frequently in plants, okay, we see errors, if you want to call them errors, we see a variation from that diploid number. And in many cases, we end up with plants that are tetraploid, okay? This is called polyploidy. Now, in animals, we do sometimes see this. It's rare compared to plants. When we do see it in animals, it's mainly in reptiles, amphibians, and fish. We do not see it a lot in mammals. We do not see it a lot in birds. But in these groups here, it does seem to be somewhat tolerated, but it's under very specific circumstances. And we're going to focus our conversation mainly on plants for this because it seems to be a normal part of their evolutionary path. It may come down to the fact that plants have a hard time moving, since most of them don't. They don't. And it might make it a way where speciation can happen without the need for geographic separation. If you're interested in that, go see some of my other videos on evolution, particularly those on allopatric and sympatric speciation. So polyploidy is an example of a way in which sympatric speciation can occur. That's speciation without reproductive isolation, or I should say geographic isolation, because they will eventually be reproductively isolated simply because when you have a different number of chromosomes than other individuals, it's very hard to make functional offspring. But let's talk about this. So there are two different types of polyploidy. There is autopolyploidy and there is allopolyploidy. Now, auto should trigger in your head that this is going to happen from within the same species. So this is when you get additional chromosome sets that arise from the same species. Allopolyploidy is what happens essentially when you crossbreed species. You often get individuals with different numbers of chromosomes, but somehow they fuse together to form a hybrid that has a different number of chromosomes than either of the parent species. What's interesting is through either of these mechanisms, the resulting offspring essentially becomes a species of one, because in either case, you are going to have offspring or progeny that have a different chromosome number than their parents, even if it's their parents' fault. So in autopolyploidy, what happens is there is an error during gamete formation where you end up with a meiotic error where you get unreduced gametes. So let's take a look at the karyotype of this parent species in this example. Their diploid number is 6. They have three pairs of chromosomes, okay? A meiotic error, not mitosis, meiosis, a meiotic error occurs where they don't reduce. So maybe they don't do that second round of separation. And as a result, the gametes are diploid. But then these diploid gametes fuse through self-fertilization. And now the result is offspring that has 12 chromosomes instead of 6. So this likely results from the fact that plants can often self-fertilize. All right. Many plants have the ability to produce both sperm and egg. And when I talk about plants, I'm talking about the individual plant itself. Like this plant is producing its own sperm and its own eggs. And while it would be more beneficial to that plant to send its pollen off and fertilize the egg of a different plant within its species, sometimes self-fertilization, self-pollinization occurs where some of that pollen goes in and fertilizes the egg of that same particular plant, that individual. There's nothing wrong with that. That's perfectly normal. In fact, if you go back to Mendel, one of the things that worked for him was that Pisum sativum, the pea species he was working with, is self-fertilizing. That allowed him to do internal crosses where he could pollinate these plants with their own pollen to get that next generation and know exactly who's pollinating whom. All right. But in this case, you have an individual who accidentally, through an error in meiosis, produces diploid gametes. Then, because it can self-fertilize, the pollen can go on to fertilize the egg within that individual. And then, as a result, boom, you have an instantaneous polyploidy. That new zygote now has a tetraploid number of chromosomes. Now, first off, it would have to pass the viability test. This particular species would have to be like, yeah, I guess I'm okay having 4 copies of each chromosome. That needs to be compatible with life. And then that needs to reach reproductive maturity. The problem is this. Now that it's tetraploid, these gametes are likely not going to be able to fuse back into its parent plant or any other plant of the species from which it has arisen, which means basically this plant is now a species of one. But because it's from a self-fertilizing species, it can probably go on and fertilize itself. So when it goes through meiosis appropriately, it will reduce the tetraploid number down to diploid. And now moving forward, those diploid gametes will have to fuse with each other to make the tetraploid individuals of future generations. But as it produces offspring, it begins to expand, and now you have more and more individuals capable of doing this, and boom, you have an instant new species. This is how sympatric speciation works. So allopolyploidy is a little bit trickier. It's less common simply because it needs certain things to happen that are less likely to occur than something like this. But it basically happens when additional chromosome sets come from different species. So basically, a normal gamete from one species has to fertilize an unreduced gamete from another species, forming a hybrid. Now, you can see that this is probably rare because usually chromosome sets are designed to have the same number and they become haploid together and so on and so forth. But again, plants are a bit weird. They seem a little more tolerant of abnormal chromosome numbers. So let's take a look at what this looks like. So here we have species A. Its diploid number is 4. So it has 2 chromosomes or 2 pairs of chromosomes for a total of 4. Species B has a diploid number of 6. So it has 3 pairs of chromosomes. In this case, species B undergoes normal meiosis. It produces a gamete with 3 chromosomes, a haploid gamete. This particular species A has an error like we saw above. It doesn't reduce its gametes, so it now has a diploid gamete with 2 pairs or 4 chromosomes. But for whatever reason, when the sperm and the egg fertilize each other, they get this hybrid chromosome, this hybrid zygote with 7 chromosomes. Okay. Then what happens is another round of fertilization where species B fertilizes again this hybrid. Okay. Now, here's where this sort of rectifies itself. This normal gamete, okay, then fuses with the 3 unpaired chromosomes from the original mating. And now you have 1, 2, 3, 4, 5 diploid chromosomes, or 5. You have a diploid individual with 5 chromosomes. You've basically added them all together at this point. So you can see why this is a little more rare. You need this to happen and then be like, okay with it. And then species B is like, yeah, I'm going to take another bite of that apple. I'm going to try to refertilize one of these gametes. That's when the pairing happens. And now if this can undergo normal meiotic division, where they split and all that, now you have a whole new species. And again, you can see why this is an entirely new species. There's no way that this is compatible with either of these guys any longer. And now you have an instant new species. And there is evidence in nature that both of these things have happened within plant species. Okay. Also, very interesting way to check the evolution of species, because you just go, okay, whose chromosomes are these? And whose chromosomes are these? Well, this must have happened through a hybridization of these 2 species. All right. So the next thing that can happen, and again, we're getting back into conversations about how evolution happens. So I'll again refer to you, in this case, to a video of mine on the evolution of complex traits, which often result from gene duplications. And in this video, what I'm talking about is how do these gene duplications actually occur? So gene duplications are important evolutionarily because they can lead to gene redundancy. All right. What that means is that you will have multiple copies of a given gene within an individual's genome. Why that's important evolutionarily is simple. As long as one of those copies retains its normal function, the other copy can get mutated as much as it wants. And often this can lead to some pretty interesting impacts over evolutionary time. For example, you could have that second copy become a very specialized version of that original gene. They might divide and conquer, right? The gene products, you do this part of what you used to do, I'll do this part. You might have it become you might have neofunctionalism. This duplicate copy might take on an entirely new function. It's probably similar to the original function, but this is, for example, this is how a lot of our olfactory receptors came to be. You get these gene duplications and, well, this original gene used to smell something. Now they split and they can smell 2 different things, right? So how does this actually occur? Well, this occurs when we get a mispairing during the tetrads during the meiotic synapse. So you can see this would be during prophase 1 of meiosis in the formation of a gamete. You can see in this case, they weren't aligned properly. So when crossing over happened, this particular version of the chromosome during crossing over lost gene C. Chromosome 3 here added a copy of it. All right. So we get one chromosome with a deletion, one with a duplication. Now, what you might be thinking is, well, like that's probably not good. Yeah, sure. But remember, each of these chromosomes is going to end up in a different gamete, right? These are going to result in 4 different sperm cells. So yeah, if this sperm is the one that fuses with the normal egg, probably not going to be compatible with life and it just goes away. But if any of the remaining 3, in particular this one here with the duplicated C chromosome, C gene, fuses with the egg, it could be either way, sperm or egg, fuses with the egg. Now all of a sudden you have that duplicated gene carrying over. It's probably going to be compatible with life and it's probably going to be fine. We can look at how these dosage effects can cause phenotypic changes. So again, having 2 copies of the gene is going to mean in total they're going to have 3, right? So there might be a dosage effect. This gets studied a lot in genetic model organisms. One example of this is what happens in fruit flies with a gene called BAR. So this is what wild type looks like when you have 2 copies of the gene. If you take a chromosome that has a duplicated BAR gene and you have 1 chromosome that is duplicated, one that is not, you get a heterozygote for the BAR. And you can see that you start to see these bifurcated eyes. It has almost like a heart shape to it. And then if you end up with an individual's homozygous for that duplication, so in the end, they have 4 copies of the BAR gene, you get this wildly malformed eye. Okay. Oh, sorry, I went a little bit too far. So we know that these gene duplication events have contributed to the evolution of new genomes and gene families. For example, SRGAP2, if you get a duplication there, you get enhanced number of dendritic spines for the neurons. We know that a number of our T cell receptors and major histocompatibility complexes, the variation we see in humans is the result of a gene duplication. Hox genes are a big one. These are transcription factors that are involved in developmental timing and patterning. We know that there has been a large number of gene duplications throughout the evolution, particularly within animals, where we see that the number of Hox genes correlates very strongly with the level of complexity in the body plan. And we've also seen this with hemoglobin genes. So these are all examples of where gene duplications have played a role in human evolution. We see these there. So gene duplications are very important. Again, if you're interested in the context of this, go see my video on that. But for those of you who have, now you understand where these gene duplications come from. And remember, yeah, you're going to end up with a broken chromosome that's missing a copy of that gene. I mean, yeah, if you're the zygote that ends up with that broken copy, well, the end of your story is not good. But remember, 3 out of these 4 chromosomes, 3 out of the 4 sperm or 3 out of the 4 eggs that result from this are going to be perfectly viable, and this one might have the capability of leading to an evolutionary advancement. So another type of error that can happen is called an inversion. Okay. So this occurs when breaks form in a chromosome, and the region that where the break is gets recombined in an inverted fashion. Okay, so there are 2 different types of inversions. There are paracentric and pericentric, and what we're talking about is whether or not the centromere is part of the inversion. So in a paracentric inversion, the breaks in the chromosome occur. The piece gets flipped around and gets reinserted. So you can see this being shown right here. But notice that the centromere is not involved in it. Okay. Pericentric inversions, the region shoulders around the centromere. So the centromere location is also impacted by this. And whether the inversion is para or pericentric is going to impact what happens during this inversion and how this can impact things like crossing over and what happens in the next part of the process. So let's talk about what happens. So when breaks form in a chromosome, Okay. It can cause issues during meiosis when you have these inversions, because synapsis now requires what are called inversion loops. Okay. So normally during crossing over, the two homologous chromosomes that will pair off and cross over will line up like side by side, and everything will be where it needs to be. But you can see here we have a normal chromosome and a chromosome with an inversion. Now, in this case, we're looking at a paracentric inversion. We know this because notice that the centromeres are still lined up, and the impacted genes, BCD, DCB, do not go across the centromere. But because crossing over requires that homologous portions of the chromosome align, in order to make this happen, this inverted chromosome literally has to loop around itself so that B can line up with B and D can line up with D, right? So you can see there's almost like this fulcrum right here around C. And then what happens if you get a single crossing over point between B and C in this case, you can end up with a break, all right? Because the crossing over happens, but then you don't have like the compatible regions to reconnect the right way. And all of a sudden, now you have that normal chromosome and you have the inverted chromosome, but where the crossing over happens, you get basically what's called a dicentric bridge. Notice that you have both centromeres on both chromosomes are now touching each other. And then you have this acentric fragment where there is no centromere. The problem with this is that when they go to separate, when they segregate during anaphase 1, you're going to end up with a normal product and an inversion product. And in both of these cases, all genes are present. So, barring some other like issue with where that particular inversion happened, these are probably, this one's certainly compatible with life. It's remember, these are 4 different sperm or 4 different eggs, right? This is fine. This is probably fine too, for the most part, because you've got all the genetic information. But because you lost this acentric fragment, you're missing huge hunks of genes, right? So these deletion products that result from that crossing over, the loss of the acentric fragment, the random break in that dicentric bridge, now you've got one sperm that just has gene A from that chromosome. Another one has A, B, C, D, but neither of them have E, F, G, H. These two right here are not going to be compatible with life, okay? And it has to do with the number of centromeres that are going to happen when this occurs and the formation of this loop. Let's look what happens then in the example of the pericentric version. So again, this is going to involve the centromeres. So when this happens, again, we've got the normal chromosome, the inverted chromosome. Notice now that the centromeres are no longer lining up. Again, we're going to have this loop have to form so that B can cross over with B, D can cross over with D. This time we're going to have, this time the centromere is going to be outside of this region where the crossing over is going to occur because of its location. And as a result, we will get this normal gene product, okay, which is viable. This is the normal chromosome that didn't cross over. We have chromosome 3 that's inverted, but again, all genes are present, didn't participate in the crossover. But again, we have 2 inviable chromosomes. Why? Because essentially this one contains a duplication and a deletion, right? So this one gets gene A, B, C, the centromere D, but then it gets A again, right? Because of this loop and where the centromere is. And then we have another problem here where we have a deletion duplication. This one has a bunch of genes, but it's missing A altogether. So it's got a duplication. So it's also missing D, by the way. D's here. So it's missing A altogether. So it has 2 copies of H, 2 copies of G, 2 copies of E, one of B, C, and D, but A is completely missing. So it's a deletion duplication product. This is a deletion duplication product here, non-viable here. These 2 are perfectly viable. So what you're getting is different gene products because of where that centromere is located. whether it's involved in the inversion or not. So you can see again how inversions can be very, very destructive. And the main reason why the inversion is destructive is, at least during meiosis 1, during crossing over, you're going to get these insertion deletion products if it's involved, or you're going to get deletion products if it's a paracentric inversion. Okay. Again, you do end up with some viable chromosomes. And I do want to point out here that just because this chromosome is viable because it has one copy of each gene and it does have a centromere, there may be other issues. It depends on where the inversion occurs. You could disrupt a promoter that silences a gene. You could disrupt some other regulatory element in there, and you can end up with problems. So, for example, there are inversions that result in cancer because you get the wrong promoter upstream of the wrong gene and that kind of stuff. So viable does not necessarily mean healthy. I think it's important to note that. Viable just simply means the fetus will result in a live birth. Doesn't mean how long that life will be. It does not tell you what the quality of that life will be. Okay. Then we get translocations. So translocations occur when segments of chromosomes switch places. Usually you have to have a break in two chromosomes that are nearby each other, and then you end up with this weird cruciform structure, this what we call a cruciform configuration during meiosis to allow crossing over to occur. So you can see normally you would just have the two homologous sets of sister chromatids coming together to do this. But because there are parts of various chromosomes on the wrong chromosome, now they have to pair up in a weird way to make crossing over happen. So you can see the cross shape that's being formed for these junctions to occur. And then what happens depends on essentially what happens, right? It comes down to random assortment. So you get what's called alternate segregation or two different forms of adjacent segregation. So again, we're talking about now what happens during anaphase, what happens following mitosis during random assortment, because you're going to get these crossing over events and you're going to get some weird things because remember, you have part of chromosome 2 on chromosome 1. This isn't the crossing over where dad's copy of chromosome 1 is crossing over to mom's copy of chromosome 1. This is literally part of my chromosome 2 is on chromosome 1 and part of my chromosome 1 is on chromosome 2. And now they're also doing crossing over. So you can see there's a lot happening with these translocations. Now, in the case of alternate segregation, essentially what happens is when they line up on the metaphase plate, the two, quote, normal copies go to one daughter cell. Remember, this is meiosis 1, so there's going to be a subsequent round. And then the two translocated copies go to the other gamete or go to the other cell, okay? The reason why this is important is because in this case, there are translocations, but they are balanced because during the next round of meiosis, during the next metaphase, metaphase 2, one copy of this chromosome 2 is going to go, one copy of this chromosome 1 is going to go. And even though the information is on the wrong chromosomes, it's there. And it's there in the right quantity. So essentially, you do have all of chromosome 1 present. It's just some of it's on a chromosome 2. And you do have all of chromosome 2 present. It's just that some of it's on a chromosome 1. But because the right information is there in the right dosage, these are called balanced translocations. You're going to get a couple of perfectly normal wild type gametes here because of the untranslocated chromosomes. And then you're going to get a couple of balanced translocations where the missing information from chromosome 1 is found on 2 and vice versa. Same here. In the end, this is probably going to be okay because everything's balanced out in the gametes. Now, what happens in the next round, if these balanced translocations are the ones that go on to produce the next offspring, there could be issues. But if we go with the adjacent form of segregation, again, this is random, right? Random assortment or during metaphase 1, if we end up with one wild type copy and one translocated copy going in either case, now we have adjacent segregation. Okay. And what you can see here is Now we have too much information from chromosome 1 present and not enough of chromosome 2, right? There's no balance because this set of chromosomes, these pair of chromosome 1s are all chromosome 1. This pair of chromosome 2s, these sister chromatids, don't have all the information. They don't have all the information chromosome 2 is supposed to have because part of those chromosomes have information from chromosome 1. And then it's just the opposite here, right? Here's all of chromosome 2 and a little bit more. And here's some of chromosome 1, but the rest is missing. Okay? And as a result, all 4 of the resulting gametes are going to be unbalanced. This is not a good situation. These are not likely to be viable because they don't have enough information from one of the chromosomes and too much from the other. This is called adjacent 1 because you still have a chromosome 1 and a chromosome 2, a chromosome 1 and a chromosome 2. Adjacent 2 segregation, which is exceedingly rare, but can happen, is when essentially you get both chromosome 1s going one way, both chromosome 2s going the other. So while you do have a bit of chromosome 1 information here, there's a whole lot of chromosome 2 happening. You have a bit of chromosome 2 information here. There's a lot of chromosome 1. Again, very unbalanced here, and you can see the resulting gametes. So this is why translocations are problematic. Let me give you an example of a translocation, though, that is actually tolerated in humans. So this is what is called familial Down syndrome. It accounts for about 5% of cases of Down syndrome, and it results from something called a Robertsonian translocation in one of the parents. And to be clear, the parent will likely have no clue that they actually have this. Turns out that this is actually more common than we actually previously had thought. This is more common than you think it would be. So what you have to recognize, first we have to discuss what's a Robertsonian translocation. So Robertsonian translocations happen between 2 acrocentric chromosomes. The way this typically happens in humans is with chromosome 14 and chromosome 21. So if you recall, acrocentric chromosomes are chromosomes where the centromere is very far from center. Okay? If you look, chromosome 14 and chromosome 21 have a very long arm and a very short arm. You can draw them a bit like this. And in a Robertsonian translocation, basically, the long arm of chromosome 21 and the long arm of chromosome 14 fuse to make a super long chromosome. And then you have these 2 little short arms that fuse together and essentially go away. They basically don't exist in this individual. They do go away because they're so small. They basically get lost. They actually can't attach because they don't have a centromere. They can't attach to the spindle, basically. Okay, so they don't separate. They just go away. But the individual who has this Robertsonian translocation usually has no phenotype whatsoever. And the reason why is they have pretty much all the information of chromosome 14 and chromosome 21, but they are actually 45 minus 21. They are essentially missing their chromosome 21. Okay? Because the bulk of chromosome 21 is carried there. Now, what happens next depends on how does the separation occur during meiosis in this individual. Because when they undergo meiosis, okay, and we do random assortment during meiosis 1, we can get some weird effects. Okay, so we have this individual with the Robertsonian translocation. Now what we have to figure out is what happens during gamete formation in the next individual. So this is what the parent's chromosomal arrangement will look like prior to gamete production. Then they're going to undergo meiosis and they're going to undergo the separation of the gametes. So one thing that can happen, first off, during random assortment, normal chromosome 14, normal chromosome 21 go the same way. The resulting gamete is perfectly normal. One wild type copy of 14, one wild type copy of 21. And then when it fuses with a normal gamete from the other parent, you get an individual that is normal. Okay? Or you could get this mishmash of 14, 21 going one way, Okay. So again, this is the opposite of this. If good 14 and good 21 go one way, then it's this combined Robertsonian chromosome that goes. Again, balanced carrier. When this fuses with the normal gamete from the other parent, normal 14, normal 21. And as far as the body's concerned, okay, I've got all the information from 14 and 21. We're fine. Again, this will be a balanced carrier. It will carry that chromosome, and that could show up in the next generation, familial. But this individual's fine. Sometimes you get 14 and the translocated chromosome going one way. Then when it fuses, you get what's called a translocation trisomy. So you've got chromosome 14, Robertsonian chromosome, and then 21. This is too much. This is called a translocation trisomy. This is lethal. Or you get just chromosome 14 and 21 going, and they only get only chromosome 21 goes in this case. So again, in order for this translocation to happen, only chromosome 21 goes. This is a monosomy 14 or monosomy 20 or 14. No good. This is not compatible with life. These are lethal. Another scenario, again, these are all the ways that we can get random assortment during meiosis. We end up with just 14 going, right? And then the translocated chromosome and 21 go the other way. In this case, this is monosomy 21, still lethal. But in the other half of this, where the Robertsonian 14, 21, and the 21 goes, this is what's called translocation Down syndrome, because they're going to get a normal copy of chromosome 21, the Robertsonian copy with 2114, and the additional chromosome 21 from the other parent. And now all of a sudden you have what's called translocation Down syndrome. The reason being is essentially, as far as the body's concerned, there are 3 copies of 21 present. That's trisomy, and 2 copies of 14 present. And that's normal. Okay. So we have to look at it. This is basically the different ways that a chromosome, a karyotype such as this can separate during meiosis. And here, if it happens this way, you get either a normal sperm or egg and a balanced carrier sperm or egg that can result in a normal or a balanced carrier offspring. Here, if it divides this way, both outcomes are lethal. Why? You either end up with translocation trisomy or monosomy. Neither of these is acceptable. If it goes this way, one outcome is monosomy 21, lethal. The other is translocation Down syndrome. So you can see, while this is perfectly fine in the parent, what happens during random assortative meiosis dictates the potential outcomes for the offspring of this particular individual. And again, we go down to just probability, sheer luck when it comes to this. Okay, so at this point, we'll be done with the different chromosome numbers, and we're going to talk about something slightly different. We're going to talk about non-nuclear inheritance. We're going to talk about how inheritance of mitochondrial diseases happen. So, and this also happens with chloroplasts, but again, that's plants. We're not plants and algae at this point, but just keep in mind what we're talking about here can also occur in organisms with chloroplasts. But I'll focus in this particular video just on mitochondria, since it's a little closer to home for us as humans who do have mitochondria, but don't have chloroplasts. All right, so things we have to understand. Mitochondrial genes that are important for producing things in the mitochondria, whether it's a mitochondrial protein, tRNA, something like that, exist both in the nucleus and the mitochondrial DNA. But what we're talking about here are just those mitochondrial genes that are in the mitochondrial genome. So things you have to understand, the mitochondrial genome is very small. All right. It does vary among species, but for the humans, our mitochondrial genome is very, very small. And every gene encoded in our mitochondrial genome is considered to be essential. If you lose any one of them completely, it's lethal. Okay. So in our mitochondrial genome, there are 35 genes. 2 of them encode tRNAs, 13 encode proteins. That's it. Pretty much everything, and some ribosomal RNA. So there's a little bit more than that, but ribosomal RNA, okay, in there as well. Every other RNA, protein, whatever, involved in mitochondrial function comes from the nucleus. But we're focusing just on these genes, these ribosomal RNAs and these tRNA and protein encoding regions. The thing we have to understand is that when we end up with mitochondrial mutations, the phenotypes can vary greatly. And here's why. Each of our cells contains thousands of mitochondria. And mitochondria, a single mitochondrion can have anywhere from 2 to like 10 copies of the mitochondrial genome. And what that means is that because it's not like the nucleus where each cell has one nucleus, and in that nucleus, you have 2 copies of each chromosome, mitochondria can have 2, 7, 10, a dozen copies of the mitochondrial genome. And some may contain that mutation and some may not. So you end up with something called heteroplasmy. Here's the other thing that happens. Mitochondria are constantly fusing and dividing. It's called fusion and fission. Fuse, divide, fuse, divide. And whenever they divide, you can think of it as a sort of a compartment. And when they divide, whatever mitochondrial chromosomes are in this particular mitochondrion that go away, that's there. Whatever's in this one, that's its genome. And during cell division, we have to partition our mitochondria, which means, theoretically speaking, if you have this heteroplasmy, if you have some mitochondrial chromosomes that are damaged and some that are fine, well, the cell, when the cell divides, there's just this sort of random inclusion of mutant and wild type mitochondria. So you could end up with a daughter cell that just gets a truckload of mutated mitochondrial DNA and another daughter cell that ends up with the majority of wild type DNA. Or it could be a 50-50 partition. It's completely random. But if you end up with a cell that has a bunch of mutated mitochondrial DNA, that cell's not going to function appropriately. If you end up with a cell that has mostly wild type mitochondria, it's going to function pretty close to a normal cell. If you get one that's 50-50, you're going to get some mitochondria functioning in that cell and some that aren't functioning very well. And as a result, mitochondrial diseases don't take on a unique, they don't take on like a consistent phenotype. They can vary widely among individuals who have them because it depends on what cells contain the mutated mitochondria, how many of them, what cell types they are, what function those cell types perform. And as a result, you can end up with just sort of this very mosaic appearance, if you will, in terms of like, yeah, a lot of people have this mutation, but they all have totally different severity with respect to mitochondrial disease because it just depends on which mitochondria and where they are who have this mutation and how many. So our mitochondrial DNA is actually quite vulnerable to mutations. One of the reasons why is, remember, it is prokaryotic in nature. It's small, it's circular. There are no histones, so they lack protection. Because mitochondria are essentially little tiny bacteria, they don't have the DNA repair mechanisms that our nuclear genome has. And more importantly, they sit in the seat of reactive oxygen species production. Reactive oxygen species like superoxide, hydrogen peroxide, all of these things can damage DNA. So you have this relatively unprotected DNA with very limited capability to repair itself sitting right next to a mutation factory. It's inside the mutation factory. This isn't a great plan, but this is what evolution has given us eukaryotes. And as a result, we can see these things develop throughout an individual's lifetime. Because remember, our cells can't make new mitochondria. They rely on existing mitochondria to make more of themselves. And when they do that, they're going to partition their DNA, good, bad, or otherwise, into those daughter mitochondria. So inherited mitochondrial diseases exhibit the following patterns. First, it's always a maternal inheritance pattern. It's not Mendelian. Remember, all mitochondrial DNA is inherited from the mother. You're going to often see impacts on energy production because these mitochondrial genes are all involved or almost all involved in the electron transport chain. You're going to see impacts on energy production. All right. And when you see all of these, it's likely it has to be the result of a mitochondrial gene. A mitochondrial disease has to impact a gene that's important for the mitochondrion. Okay. They can affect many tissues, organs, and organ systems, particularly those that are very high in terms of energy consumption, particularly things like neurons and muscles, because they rely so heavily on the energy produced by mitochondria that defects in energy production caused by these mutations are going to be problematic for those tissues more so than others. So let's look at a few of them. The first one is called myoclonic epilepsy and ragged red fiber, also known as MRF, for obvious reasons. Symptoms include ataxia, which is lack of muscle coordination. People can't use their muscles appropriately. Deafness can happen. Dementia is very common, and epileptic seizures. The way this is clinically diagnosed is a normal muscle cell will look like this. Here you get these blotchy skeletal muscle fibers. And the more of these ragged red blotchy skeletal muscle fibers you find, the more damage is being done to the particular muscle cells in these individuals. These have been almost exclusively tracked to the mitochondrial gene, which encodes the mitochondrial lysine tRNA. All right. Again, what we see is the mitochondria in individuals who have these mutations are heteroplasmic, which means some of their mitochondria don't have the mutation and function perfectly fine, and they'll have normal muscle cells, whereas mitochondria in cells where the mitochondria are heavily damaged by this mutation, you start to see these ragged red fibers. Another example of this is called LHON, so Leber's hereditary optic neuropathy. It's a very rare disease. It predominantly shows up in people between the ages of 27 and 34, and it leads to blindness typically within a year of symptoms appearing in about 80% of patients. So it happens dramatically, okay? Basically, what we find is about 50% of the mutations that lead to this are in a complex 1 subunit. All right. About 50% of these mutations occur in a subunit of NADH hydrogenase known as complex 1. And as a result, their cells are not able to perform OXPHOS at the rate needed, and it results in decreased energy production. What's interesting here is there does not seem to be a familial link with this. This appears to be something that happens sporadically. Individuals develop this mutation, and then within a year, they're about 80% of them are completely blind due to this particular defect. So this is not something that's typically inherited. This is something that's going to happen just spontaneously in an individual. We still don't fully understand this disease, why it happens, and why the age of onset is usually around 27 to 34 years. We're still learning more about it. And then the last one we'll talk about is something called Carnes-Sayre syndrome, abbreviated KSS. Symptoms of this include vision loss, hearing loss, and heart conditions. And what we've noted is that this involves deletions at various positions in the mitochondrial DNA. What's interesting is many patients who have this are symptom-free as children, but as they get older, they see more and more of the problems associated with this. And essentially what we see is as they age, we see increasing numbers of mitochondria with the deletions in their mitochondrial DNA. And as more and more of these accumulate, the symptoms get worse and worse. Okay. So I hope this gives you an idea of some of these mitochondrial diseases and understanding that how mitochondrial diseases get inherited and how they occur. The big thing with these is it comes down to heteroplasmy. So here's kind of how they're talking about this with Carnes-Sayre syndrome, but this is a good example for what can happen in any of these inherited mitochondrial diseases. Here's the mother with mild or no symptoms. These green mitochondria are healthy ones. These orange ones are the ones that are defective. And then when mom goes to produce the eggs or the cells that will become the eggs, the mother cells here are going to partition their mitochondria. And some of these mitochondria or some of these cells will get lots of affected mitochondria. Some will have very few, and some will have about a 50-50 mixture. Depending on which of these then go on to become the mature egg that gets fertilized, this is going to lead to the chances of or how profound the illness might be in the child. If there are lots of mutant mitochondria, the child is likely to have severe forms of the disease. If it's a 50-50 mixture, it's mild. And if there are very few of those mutated mitochondria, it could be a child with no disease. Again, we're still learning to understand more about these mitochondrial diseases, but this is the current thought process on how this happens. All right, that's it. Thanks for tuning in. I hope you learned a lot, and I will talk to you all real soon.